Good morning. Thank you for the organizer for inviting me. Today I will talk about the work in the Fundación Instituto de Inmunología and the Escuela de Medicina of the University of Rosario. In my group, the biomathematical group, we study the interaction of peptide with the MHC molecules. This is the aim of our institute is develop vaccines for infectious diseases. We are devoted in the study of malaria vaccine, and malaria is still a very important cause of disease and death in the world. The problem is design peptide-based vaccines, and we have an interdisciplinary approaches to accomplish that goal. The first is the use of information based on the polymorphisms of parasites to develop peptides, conserved peptides that were critical in the function of the pathogenesis. That conserved sectors, frequently is associated with binding properties to host cells, but also for interaction with another kind of proteins of the parasite. This in immunological activity of the peptides, it's so bad. It frequently is silent and undetectable for the immune system. So some changes in their sequence provokes that peptides turns to immunogenic and sometimes protective. The reason of this is the increase in fitting to the major histocompatibility complex. So the general strategy of FIDIC comprises all these parts. The first is the protein identification of vaccine candidates based on function, pathogenesis, and binding properties. The other is the variability patterns. That variability patterns we focus in conserved sequences because we determine that conserved sequences could have a functional restriction, and that point to critical regions. Another criteria is the structure of these peptides and its degree of exposure to immune system. Another part of the strategy is the design of peptides based in amino acid characterization and preserving folding properties of these peptides. The part of my group involves the determination and use of structural and information to the fighting of peptide in the MHC and its relation with the T-cell receptor. Another kind of line is the modification of that peptides that increase the immune activity, the immunogenicity, and as a final goal, the protection. The final part is this development of vaccines and involves the use of animal models, the use of characterization of major histocompatibility profiles and their equivalence with human populations. Also the characterization of human population and the MCC profiles and the coverage or potency coverage of this peptide design in the population and the effectivity for the vaccine. Another kind of line is the evolution of the resistance of these vaccines in itself. So the key of all this story is the molecule of the major histocompatibility complex. The major histocompatibility complex constitutes an important part of the immune system. During infection, pathogenic proteins are processed into peptide fragments by the antigen processing machinery. These peptides bind to MHC And the MSC peptide complex is then transported to the cell membrane, where it elicits an immune response via T-cell binding. These molecules are very complex in the sense of constitutes a multi-allelic family. We have major histocompatible complex class 1 that is involved in presentation of endogenous antigens, and major histocompatibility class 2 that is involved in the presentation of exogenous antigens. We focus in these kind of molecules because our interest in the parasitic diseases. So the characteristics of these molecules comprises a complex evolutionary mode, a strong selection pressure, and a high polymorphism as response of the high diversity of the different pathogens proteins. So in the modes of the evolution, it comprise all these factors, and the main factor is the coevolution with the protein of pathogens. And this molecule is critical not only for immune function, but also for effects as sexual selection and maternal effects. We use as animal model the primate Aotus, an Amazonic monkey, and we determined that there exists a molecular and selective convergence between humans and Aotus. In this case, in this work, we prove the narrow relation among these molecules in Aotus and human over other counterparts in more similar primates to human beings. This polymorphism it's very great. In the case of the molecules of especially studied, it is the HCDRB comprise hundreds of different alleles. So these polymorphisms, in the case of human populations, could be characteristic for each population. In this case, we have a profiles of the different lossy lineage for these alleles, and these could be used, looks very different among different populations, but inside the kind of population like Amerindian, the variation is also high. So in an analysis of the diversity of the different populations, we can determine three main groups to design peptides. One comprise population of Africa and Europe, Asian, and Native American people. So the structure of the DR is in class 2 is paramount for the design of the peptides candidates to vaccine. In this case, I showed you the immunological synapse, and the immunological synapse involves the interaction among the T-cell receptor and the major histocompatibility molecules mediated by a peptide recognized pattern. And this kind of recognize, it's very specific and comprise the interaction with a subreceptors named pockets. The selective pressure produce as variations of a single theme. The structure of these molecules is very conserved in spite of the huge diversity, but this diversity is differentially concentrated in residues involved in backbone interactions. That residues used to be conserved and are submitted to negative pressure selective pressure, and the variable residues often interacts with the side chain of the peptide and constitutes the nucleus of the reconnaissance and the binding process. So, in our institute, the study of the structure points that this structure, the pi structure, the extended structure, it's critical to the proper binding process and activation properties. So we use crystallographical base and try to design replacements to modify the structure of peptides candidates to vaccine. We use the protein geometry database and make a bioinformatics approach to determine the Ramachandran distribution of the different amino acids and determine the amino acid structural propensities. And for our surprise, these structural propensities corresponds very well to Blossom 62 matrix, point to mutational basis of alignment in proteins. We have a strong correlation with the different mutational matrices, and the mutability itself is related with secondary structure and mass. We also try to determine the binding properties of the peptides candidates to vaccines, and this process of PMMC binding corresponds on understanding the molecular mechanism of MH binding correspond a great advantage to the design to effective drugs and vaccines. An empirical approximation, it's very difficult because there are billions of potential binding peptides and there are thousands of molecules. So computational approaches offer the promise of predicting peptide binding, and this could dramatically reduce the number of peptides for experimental verification. So we have two main classes of approximation. One of these is the use of sequence-based approaches that use binding data to train machine learning approaches, software. And the other is the structure-based approach that considers the structural features and the distribution of energy between the binding peptide and the major histocompatibility complex molecule. The structure-based methods have a great advantage because they don't need the use of binding experimental data, since they determine the structure and the requirements of binding using a theoretical framework could constitute an adequate approach to this problem. So we use quantum semi-empirical methods. These semi-empirical methods can be defined as the simple version of electronic structure theory. Some two-electron integrals are rejected to speed up the computation. Some integrals are computed using parameters for experiment, using semi-empirical data. And only balanced electrons is considered. The core is treated by reducing nuclear charge or by adding special core functions. This is the methodological step that I use. We use a structure from PDB, make mutations in the program Chimera, make up approximation process in the program MOCPAC, we have an optimization in MOPAC, and we measure the binding energy using this semi-empirical method, the FMEOM, the FTP in games, and the binding energy in using PM7 and Cosmo in MOPAC. In this work, we use two sets of data. One set of data consists in the hemagglutinin peptide and a series of monosubstitution for aspartic acid. And another set is the use of myelin peptide using a panel of monosubstitution by alanine. In this case, we found a very good correlation with the binding IC50 parameters, and also very good sensibility of this proof. And in this case also, but it's clear that the use of DFTV could be better or approach to establish the binding properties of peptides. So in the next part of this work is use an automatic setup, and this pipeline take more consideration among them, considering the neutral and charged states of the proteins, considering solvent effects and considering rotamer conformation effects. We use also this hypothesis that the enthalpic effects, its energy, it's more greater than the entropic effects, and we have empirical evidence of that recompiled for 4 other groups, but very conclusive respect. We use also 3 semi-empirical quantum methods, PMCs, PM7, and DFTV with fragmented molecular orbital approach. We use 4 sets of peptides with different receptors. We have a very good time of computation for the different experimental setups. And we have a very good Pearson correlations with DFTV here. Here we have PMCs and PM7. And this is the gold standard in prediction. It's a neural network predictor. Also, the approach, it's very precise. This is the root mean square deviation. We note that the experimental error is near those 2 kilocalories per mole. And we have a very good performance for DFTV methods in the 2 flavors, neutral and charged. We have one of the sets, the correlations in this case, for DR1 with HA peptide, DR1 with CLIP peptide, with DR2 with myelin peptide, and DR3 with CLIP peptide. In all cases, our approach is very precise for DFTV, not so good for another methods like PMCs and PM7. And the outlook of this work is we will extrapolate our methodology to other peptide major stoichiometry complex systems, including those with unknown crystallographic structure, but which can be easily predicted by homology modeling. And our proposal can also be valuable for calculating the binding energy for systems having similar structural boundaries. So as a synthesis and a part of our approximation, we have performed pocket profiles, lineage and population profiles, and binding profiles. In the pocket profiles, we construct a pocket dictionary. This pocket dictionary is based in the residues in contact with the peptide, these superreceptors, pockets, and we have here the variance of this pocket in human and here in the animal model Aotus, and we have an intersection with 4 of these profiles, but these comprise the 93% of all the kind of pocket 1 in Aotus and the 98 pockets in humans. So the evaluation of these profiles using approximation based on computational chemistry could be a base of a fast screening method to develop. The other focus is the profiling lineage. This is an attempt to deal with the polymorphism. In this case, we have that the pocket profiles integrated with the different subreceptors can offer a 60% of coverage using a small amount of allele prototypes. So the number of receptors necessary to accomplish a prediction method or a develop of peptide-based vaccine, it's very low compared with an explicit approach to the program. So the integration with these profiles with the percentage in the population offers an opportunity to develop a high vaccines with high coverage in the populations, and an opportunity to develop vaccine for other kind of disease. And finally, binding profiles in using an structural-based approach is also explored for our group using another kind of descriptors as electrostatic properties, and having also a very good correlation with experimental data. So the integration of these strategies tries to design an accurate identification of peptide binding to major histocompatibility complex molecules, and the explanation and simulation of the mechanism involved in the process is of great importance in the understanding the mechanism of immune recognition and facilitating the process of epitope-based vaccine design. Our group is conformed by William Agudelo, Hugo Borges, Ronald Gonzalez, Carlos Andres Maecha, and they are chemists. And for me, Carlos Fernando Suaréz, I'm a biologist, and my expertise is in evolution and bioinformatics. The counterpart in immunology is by the Professor Manuel Alfonso Patarroyo and the director of our institution, Manuel Elkin Patarroyo. Well, thank you.